Hey 7th graders, it's Miss Crawford. If you guys are DLI, you guys are doing this food lab at home, or if you were absent on either Monday, Tuesday if you're an A-day kiddo, or Thursday, Friday you're a B-day kiddo, this is a lab you're going to be doing for class. So if you're missing class today, this video is really important to kind of explain everything to you. So you can find, obviously if you found this video, you found the assignment, which is in Teams Assignments. In the video, there's 3 things attached that you need. First of all, this video. And also your worksheet, which looks something like this. It's because it says Food Lab at the top. And you need this PowerPoint presentation that kind of goes through the results with you. So this is where you'll get the answers to your guys' lab since you won't be doing it in class. So basically what you guys have been learning about lately is when we eat something, it breaks down in our stomachs or in our small intestines, our body absorbs it, and we use those nutrients that we get out of the food for different processes in our body, like creating energy and metabolism. With that being said, we're going to run through a couple tests. So there's basically 3 or 4 different things, however you guys want to look at it, that you guys can get out of food. You have carbohydrates, which is one of them. But when I say 4, there's 4 different things you can get out of food. There's 2 different types of carbohydrates, and I'm going to talk about those here in a second. You could also test for fat, which we call lipids in the science world, and proteins. You guys probably know proteins just about the best because you think of proteins when you guys think of meats. You probably think of pasta when you think carbs or breads, but they're a lot more than that. All right, so let's just go through. Here's the 4 different tests that we're going to use. So you'll be testing for carbohydrates, which again, there's 2 different types, sugar and starch. You'll be testing for proteins, and you'll be testing for lipids, which are fats. So here's your 4 tests. For proteins, you're going to use the Burt Test. For sugar, which is a simple type of carbohydrate, you're going to use the Benedict's test. To find a starch, which is complex carbohydrates, so these are both of our carbohydrates, you're going to use iodine. And for the lipids, you're going to use a brown paper test or a spot test. Okay, so first test I'm going to talk to you guys about is your Burt test. So this one's pretty simple. Basically, you would have a test tube with whatever substance you're testing in it. It could be something like egg whites from the lab today, or it could be something like apple juice, and you're going to test to see if it has protein in it. Okay, so a negative test, the solution itself is blue in color. So we would add the Burt to your sample. And if it changes to this lavender color, that means there's protein in that food. Okay, so if protein is present, it will turn lavender in color. It won't turn any other color. So you guys got to count, like, did your solution have a color before you added the blue? Was it just a mix of color? If it turned purple, however, though, that means there's protein. So hint, protein turns purple. Okay, so you guys can think of the little P's there to help you remember protein turns purple. Okay, Benedict's test. So let's just take a second and talk about our 2 different types of carbohydrates. Okay, so we've learned so far with Amplify that when we eat a carbohydrate, it breaks down into glucose in our body, and glucose is really important for metabolism for us. So on a molecular scale, if we were to zoom in way past a cell, way past any virus, we could see this model for glucose. So basically, you guys eat something with a carbohydrate in it, and you guys get this teeny tiny single glucose molecule. It makes this little ring shape. Okay? Sugar that you guys like maybe add to cookies when you bake them is a disaccharide. It means you have 2 glucose molecules hooked together. So you have 1 and 2. Okay, so that's sugar that you guys eat. Like I said, if you guys maybe are eating Rice Krispies and you put a load of sugar on them in the morning, or you're making cookies and you add the white sugar in your guys' pantry. So those are your simple sugars. You guys would also find these sugars in something like a strawberry. Different fruits have these type of sugars. Really quite a few food items have sugars. Okay, but when we're talking about starch, we're talking about something a little bit more complex. So this is 1 glucose molecule, this is another, and they kind of branch off and connect. They can have up to 1,000 glucose molecules in a starch. So starch is a complex carbohydrate. So it takes a little bit longer for us to digest than, say, a strawberry with this type of sugar. Okay, so pasta or bread, you guys would see a lot of this type of starch that has almost 1,000 possibly glucose molecules hooked together in a big chain. So when you guys digest this down, it takes a while, but you break into quite a little bit of glucose. If you guys know anyone who's a runner, you might hear of them carb loading. So they eat a big dinner the night before they go on a race. with like maybe a lot of pasta or a lot of bread, because it has this starch in it that slowly breaks down in our stomachs and gives us a lot of glucose that helps maybe in a marathon or whatever someone's running. Okay, so those are the difference between a simple sugar and a complex sugar or starch. Okay, so to test for simple sugars like glucose, okay, or even a disaccharide, you guys would use a Benedict's test. This one's probably the most complicated out of the four because you have to boil it. So the liquid is blue in color. You would add it to your sample of food, and then you would boil it, and then you look for a change of color. There's not a specific change of color because it depends on how much sugar and what kind of sugar is in there, but the main one that you're going to see is kind of this rusty orange and maybe even this red color. But you'll see a color change. If it's negative, it's probably going to stay this blue color, but after boiling it, if it's positive, it's going to start to turn one of these other colors. Okay? And then again, make sure if you guys are taking into account the fact that maybe your substance is brown and you add the blue to it, that's not going to be a color change. That's just you mixing blue and brown together. Okay, so you're going to notice a different color change that comes out of this. Okay, iodine test is pretty simple. You guys have iodine if you guys have ever had surgery or seen someone have surgery. Iodine is that kind of orange substance that they rub on your skin to disinfect your skin. It also helps us test for starch. So remember, this starch is this really complex chain of glucose molecules. So if you guys, so that's orange in color, so if you have a negative test, there's no starch there at all, it's going to stay this really rusty orange color. But if you guys do have starch, your starch is going to turn this almost blue-black color. So it turns really dark. So this is a positive test. That means that that substance had starch in it. So iodine is a dark rusty color. If starch is present, it will turn a dark blue or black color. Okay, and finally, to test for fats, we use a really simple test called the brown paper bag test or a spot test, where basically you take your substance and you rub it on we're going to use paper towels in this case, you rub it on a paper towel and you let it dry. If it dries normal, it's negative. There's no fat. How many of you guys have ever gotten some kind of grease on your clothes, or you've seen a napkin and it doesn't dry? It never looks dry. It always looks like it stays wet. That's basically because it has a fat present. So if fats are present, the brown paper bag will dry a translucent color. So it'll always look like it's wet even after you dry it. So it's basically just a grease stain. So you guys will be able to see that. All right, so I'm going to go ahead and show you guys your worksheet. So before you begin with any of your results or go any further in this PowerPoint, you guys are going to make predictions. So you're going to go through and say, all right, water's my first food sample. I'm going to make a prediction. Making a prediction is as simple as putting a plus or a minus sign. So do you think water has protein in it? I'm going to say it's just regular water, so it's negative. Okay, do you think it has fat in it? I'm going to say water shouldn't have fat in it, so I'm going to put a negative in there. And then finally, carbohydrates. Do you think it has starch? Again, water shouldn't have anything in it, so I'm going to put a negative in both of these because I don't think it has sugar and I don't think it has starch. So I've just filled out this table with my first prediction. Okay, I'm going to go ahead and turn these a color for you guys. Okay, with that being said, you guys are going to go through, and before you begin any of your tests, you're going to go through and say, all right, now honey. Do I think honey has proteins, fats, starch, sugar, egg whites? So go through and make all your predictions for the rest of these: egg whites, vegetable oil, flour, butter, apple juice, Sprite, milk, and peas. Once you've made your predictions for all of these, then you can go do the lab part of this. So this is your actual results. So let's go ahead and talk about water really quick. So once we do our actual results and we're done with our predictions, we can go in and look at our first sample. So our first slide after our tests is the water. So water is what we call a control. A control is something that we can use to compare all the other items to. So for instance, if we're doing egg whites, I can compare my sample of egg whites and say, what do my results here look like compared to the water? Water, we know, has nothing in it, so it's gonna show us what a negative test looks like. So you can see this is before I added burette, and this is after. Notice how it's the same color of that burette solution. It's blue. So that means that's a negative test. If you guys forget, go back and check. All right, negative test is blue. Okay? So I would go into my lab and say, all right, water had no protein. That's negative. Okay? Lipids. Okay, brown paper bag. This was before I added water. This is after. Notice how minus the wrinkles, it's pretty much dried the same. Again, if I go back and look, that's a negative test. It looks the same. Okay, so I would go into my lab and I would record negative for my lipids. All right, carbohydrates. Let's go for starch. Okay, so my first one is starch. Notice how it's still that same rusty orange color that starch looks like. So again, I can go back and cross-reference and say, all right, still that orange, rusty, red color. So it's a negative. So I would put negative in here. And last but not least, we got sugar Benedicts. So we would add the Benedicts and then we would boil this. Remember, Benedicts is also blue in color. It's this lighter blue compared to the Burette. And I can look and say, all right, that's going to be a negative test here. So it's still that same blue color. So as predicted, water is negative for all 4 of these substances, as it should be. So you guys are able to look at water and see all of these negative results, okay, which is exactly what water should do for us, okay? Now you're going to go and do the same thing for honey, okay? So honey is our next slide, and you guys can see, all right, protein. It should turn lavender, but it didn't, so that would be a negative test for honey. Okay, lipids, if I were to smear a mixture of honey on a napkin, you see a little bit, but it's not that translucent, almost kind of clearish color, so that would be negative as well. So I plug negative in there. Starch, okay, still that rusty orange color. Notice the color beforehand. Okay, so I would go ahead and put a negative for that. And for sugar, this one, you can see what it looked like before. I added blue liquid to it and see how it turned this almost orange reddy color. So that would be a positive test for sugar. So I would go ahead and add a plus sign in there. Okay, so you're going to go through the rest of these: egg white, vegetable oil, flour, butter, apple juice, Sprite, milk, and peas. Obviously, you guys won't have to clean anything because you're just looking at pictures. And then when you're done, answer these questions. Why did you use distilled water as one of your test substances? Okay, so remember what we talked about when we talked about water. What is water called and why do we need it? What does Benedict's test for? If you guys don't remember, you guys can go back and say, all right, Benedict's, what is this test for? Okay. What is iodine? What is Burette? And Burette reagent will turn your skin brownish purple. So it has this purple color if you guys get it on your skin. Why do you think that is? Okay. What do carbohydrates like sugar and starch break down into? This is basically what you guys have been learning about in Amplify lately. So if we eat a carbohydrate, whether it's sugar or starch, what do we digest it to when it goes into our body? What do we absorb? And then why do carbohydrates like sugar and starch need to be broken down in our bodies in the first place? So basically anything. Why do we need to digest anything? Okay, and then what are proteins broken down into during digestion? Again, that's from your Amplify assignments. And then why do proteins need to be broken down in our bodies? Okay, so this is for the rest of you guys. You don't need to use these, okay? This is for if you were in class testing, okay? So if you guys have any questions, please let us know. And I hope you guys are having a wonderful Monday, Tuesday. Thanks.